Folks, I'd like to talk to you a little bit about vision. And to start, I'd like to talk about the area V1, right, which is the visual cortex. And of course, that'll be back, in the ultimate receiving area in the back of the brain from the eye to the LGN and the thalamus, and then to the visual cortex for the first level of, quite high level processing. And we say that the organization is called retinotopic organization in the visual cortex, because in other words, it's similar to the retina. What is viewed like, not viewed, but what lands on the retina. So in other words, if two things are next to each other on the retina, they're probably next to each other in the area V1 of the striate cortex, right? In other words, in the visual cortex. And they're similar to the LGN, which is the lateral geniculate nucleus of the thalamus. There are 6 layers of cells that are rather distinct in the visual cortex. And I'd like to go into some of these ideas, particularly beyond just retinotopic organization, but the concept of feature detection in the visual system, because we know that we don't really see in the same sense as the eye, right? In other words, people say, well, the eye is kind of like a camera, and it kind of is, but the brain is nothing like a camera, because as we have seen, what the eye does is it sends impulses into the brain, and the eye itself is very much like a camera, because why? Because it has a lens, it has an aperture opening, which of course would be the iris, and it has the retina in the back, which is kind of like the film in a camera where the light falls on. And notice that the information goes into the brain, but notice that it's kind of decomposed. In other words, when we talk about features of visual stimuli, we can say, okay, we have in this picture here, we've got the rose, we got the color, you've got the 3 cones involved with that, and of course we have the opponent process theory for color vision. So we have color vision, and then we have color, form, and we have motion, right? So in other words, we have lots of different things to kind of assemble into a percept or a perception of a of a flower. Now, in different biological systems, take a frog. Well, people have identified different types of features that the frog is able to see. So in other words, when it sees a fly or something, some of the potential prey item or food source, the frog has edge detectors. The frog has moving edge detectors. In other words, if the edge is moving, so the frog is able to detect like maybe a snake or something like that. In other words, the frog's behavior, of course, is kind of pre-wired to respond in a non-thinking fashion automatically to different kinds of stimuli that it sees, or that appears in its visual field. Now, if we look at something like, well, here's a bottle of wine, right? Now notice that you got the Maison Castel, right? So Maison Castel. So in other words, you got lots going on, a very busy label on the bottle. And notice that this is very, very exquisitely drawn, right? In other words, look at the letter A here. Well, that letter A is composed of many features. You have edges, you have orientations of lines, you don't have much color, you just got black and white there. But notice you have very exquisite forms here in the logo for the wine company, right? So in other words, the visual system kind of decomposes the little pieces there and then puts them together into a single visual perception. So we can say, oh, it's a label on a bottle of wine, right? And people have identified different kinds of cell types in different levels of the visual system. For example, in the ganglion cells. Well, we have seen the center surround antagonistic receptive fields that are able to stimulate the ganglion cells, which make up the optic nerve that goes from the retina into the LGN of the thalamus. And now when we get up into the visual cortex here, we're in V1, visual cortex area number 1, we have simple, complex, and hypercomplex cells in the visual cortex. The simple cells would respond to to bars of light, in other words, that have a specific type of orientation. So in other words, maybe a 45-degree bar of light, or maybe another one would be sensitive to a 50 degrees orientation. And, sometimes motion is required. In other words, other cells would be involved with registering not just that bar with that particular orientation. When I say a bar, I mean a little feature, a little tiny piece of, an angle, or, an angularly oriented piece of light. Look at this letter M for the word medium here. Well, that letter M is composed of 3 bars. In other words, there's 4 bars, right? And you have a few angles going on there. You got a lot going on just in the letter M, right? Now the complex visual cells require, as we said, we not only the orientation of the line, but also movement, in a specific direction. And then the hypercomplex cells respond best to. These are nerve cells in the visual cortex. So usually people study like the cat. And we have every reason to believe that it's very similar in humans. But the kitten and monkeys, different kinds of species that people have studied. And again, this is another look at the same thing: receptive fields of cortical neurons in the primary visual cortex. That's area V1, Victor 1 in the cortex. Remember, the cortex itself is not thick; it's just a few millimeters thick, but yet it contains thousands and thousands and thousands and thousands of cells, nerve cells. Now, scientists have discovered, as we know, there are. If we look at the cortex, and we can say, okay, this is the striate cortex. This is the area of the brain that we're talking about here. The eyes would be up here in the front, and this is in the back of the brain. And so notice that this little piece of tissue from the cortex is dedicated to vision. Now, notice that there's like alternating bands that are called ocular dominance columns that are located. They're like strips of tissue. And there's several layers of cells here that scientists have identified that by probing and actually recording from these different cells at different depths in the cortex. This is going, into the cortex, from the surface of the cortex down into the cortex. Now, notice what we have here. We have lines of different orientations. All right, this line is a vertically oriented line, and the other one as well. It's not quite vertical. All right, it's a little bit tilted. And then we get a bigger tilt here, and this one may be 45 degrees, right? So you get different orientations of tilt in the visual stimulus. So what's happening here is what? Well, you're taking the kitten, the cat, whatever the species is, and the cat is restrained. It is anesthetized, but it is able to process vision. So the cat's eyes are opened. Right, and the cat is placed into its head is immobilized, in kind of a holder in the laboratory, and the scientists are presenting the cat with lines of different orientations, and they have found that different parts of the cortex are sensitive to those different orientations of lines. See, so as an electrode, for example, which is that little device that's thread thinner than a thread, that measures electrical activity in nerve cells, as that electrode is sent into the cortex, it encounters many neurons, of course, thousands and thousands of neurons, but they all seem to have an orientation in this particular column of tissue here. In other words, this orientation column would have an angle of, I don't know, maybe 50 degrees, whatever this line is here, And so as the electrode goes into the cortex, the sensitivity of the nerve cells tends to be quite specific to that kind of a line orientation. Remember, we're recording from the cat's cortex as its eye is stimulated. So this is very delicate surgery. Now, notice that in these, embedded in these, these ocular dominance columns, as they're called, are these what are called blob cells. Right? Now, color sensitive blobs is what they're called. Sometimes they're called cortical pegs. and they are sensitive. They tend to register different shades of color, as opposed to, remember that the eye has the three types of cones, the short wavelength cone, the medium wavelength cone, and then the long wavelength cone for red. But notice that when you get up into the cortex, these blob cells, they're not, they're collections of cells, and they're sensitive to specific finer shades of color. This is where the actual colors are probably registered, the different shades of red and blue and green and yellow. And so notice, as we said, these are ocularly dominant, right? So here's this L stands for the left eye, then the right eye, right? And then alternating with the left and the right eye, right? Now notice that as we, here's another look at the same thing, ocular dominance column. So as you can see, here are the different orientations of the lines that the kitten, the experimental subject has been presented with. And this column is all going to be kind of dedicated to a line of that orientation. It looks almost vertical, right? So this one here looks like horizontal line, right? And here we have the alternating banding patterns of LR, left eye, right eye, left R, right eye. This is what Hubel and Wiesel were very famous for discovering in some of our other course material, is that what this is, I don't know how, it's probably not very obvious to you. These are bands, right? These are ocular dominance bands. And notice that in a normal cat, and here's just for comparison, this is 1 millimeter in size here. But notice that what the research has found that Hubel and Wiesel discovered back, in the 1960s, 70s, is that when one of the cat's eyes, this is a kitten, kitten's eyes are sutured shut so that the kitten is unable to process light coming into that sutured, sewn shut eye. Well, the other eye tends to take over the territory on the visual cortex. This is the visual cortex up here. And what happens is that by special techniques, the scientists are able to inject like a radioactive dye. I believe this is radioactive, I'm not sure. But these are, in other words, that the dye gets taken up into the nerve cells from the eye into the LGN of the brain. And then the electrical, or I should say the radioactive dye gets taken up and it actually passes through to the cortex. So what we see here in this top trace of the two pictures is alternating bands, and notice they're equal size. In other words, those little ribbons there, well, one is white colored, one is darkly colored, but they alternate, and they refer to the left and right eye. I don't know which one is left and right. The black black is left and white is right, or the reverse, whatever, right? You see, in other words, they alternate in those ocular dominance bands. Now look what happens down here. Notice that the banding pattern for the dark color is very, very slim, very small. And what has happened is the eye that was sutured tends to be represented in the cortex by far fewer nerve cells, you see. So in other words, in the bottom picture, it's almost all white. In other words, the white bands have basically taken over the cortex, which tells us that, in general, the eye that was left open, but not sutured shut in the kitten. And then, at a certain time, maybe after several weeks, maybe a month or so, and then the sutures are removed. Well, notice that the kitten is going to be essentially blind in the sutured eye. So the eye that was open during that critical period in development of the kitten tends to be represented in the cortex by these much wider, greedy, white-colored bands. Right, it takes over more space. So it's kind of like they're territorial. So there has to be what they call like a balanced competition between the right and the left eye during development. Probably the same in humans, although obviously this kind of a study can't be done in a human. But notice that the development of the nervous system from the eye to the brain requires input to the eyes. So this is kind of a picture of how much real estate, in other words, how much territory is taken over by the two eyes. Well, it's fairly equal in a kitten that is normally, visually stimulated. In other words, one eye is not sutured shut in the top picture. In the bottom, the area for the white color represents the input or the dominance by the eye that was not sutured shut. I'm not saying that the kitten has, extraordinarily acute vision in the open eye. The point is the open eye it's normal, but the kitten is essentially blind. The eye in the eye that was sutured, even though now the sutures are removed. This doesn't work in an adult cat. If the visual system has already developed, this will not happen. But it'll happen in the development, shortly after birth. It depends on the species, for several weeks. For humans, I don't know what it would be. For kittens, it's a few weeks. So these dominance column, this is what we were looking at. Notice in this picture and in this one here, notice that the right eye and the left eye alternate on the cortex, and they get fairly equal amounts of space dedicated to themselves, right? But notice that in Hubel and Wiesel's work, where they actually sutured one of the eyes shut, these banding patterns up here on the surface, these are what we were looking at. So some of them can be greatly expanded if that eye is open, as opposed to the other eye that was sutured shut. So that's one of the important findings that scientists have encountered in vision research, the ocular dominance column idea. Now, in your reading, you'll encounter an important set of ideas about the role of color in memory. In other words, we tend to recall things that are colorful, believe it or not. If you look at a colored movie or got some colored images, you may retain them better in your memory than a black and white movie or a black and white picture for some reason. And your textbook talks about, well, gee whiz, it may have something to do with the neurotransmitter glutamate, because that involves what's called a process called long-term potentiation. Now, so we have to see. And incidentally, it works particularly if the color is natural. In other words, the color of a blue banana might not work well because it's contrary to our archetype or our expectations of banana colors, which are generally yellow or maybe some are orange. Now notice that what scientists believe is that in the first stage of memory formation, color seems to have a strong impact on mediating this color phenomenon, where it's easier to recall colorful things than less colorful things. And just briefly, remember that memory itself, we haven't talked about this much at all, if at all. Memory has three stages. The first stage is called sensory memory. The second stage is called short-term memory. And the third stage is called LTM, or long-term memory. So color would have its impact probably here in this first stage, in the sensory part of memory formation. And and that's the part that's involved with kind of drawing our attention to things. So remember that, now recall the idea of the three stages of memory. What is a memory made of? Well, memories are made of nerve cells, right? So, for example, this is called the cell assembly model of neurons, I should say, of a memory, right? And so, for example, this tells us that this picture of an apple is represented in your brain by probably thousands of nerve cells that are interconnected. Here's a picture of a pineapple. Right, here's a picture of Isaac Newton, who's famous for his observation of the apple falling from the tree. Right, so we can say that we know that memories are made of connections between nerve cells. And for example, here we see that there's this is a cell assembly, as it's called. These are neurons here, right? So we'd say, well, the direction of transmission would evidently be over here. So in other words, something triggers this visual experience. And, you know, as we said, this would be sensory memory. Now, there's one for vision, which is called iconic memory. Sometimes it's called eidetic memory. So, and the one for hearing is called echoic memory. Those are probably the most studied. So the idea is when you see something, you get a brief image and you keep it in your mind's eye for maybe, you know, less than a second, but that is enough to trigger a process of moving it into short-term memory if you notice it. So what happens is that as the nerve cells become activated, they tend to, this is the formation of a long-term memory. In other words, it's an assembly of cells, so that notice that each cell, this of course is kind of a cartoonish picture, but notice that this cell here is talking to this next cell using the neurotransmitter type of communication. And this cell excites the next cell. So there's a series of connections that are made from cell to cell. And this process is called LTP, or long-term potentiation, right? And so what seems to happen is that long-term potentiation involves the something called upregulation, which means that the neurotransmitters are becoming more effective. So notice that in the process of, and there's a whole bunch of chemistry involved here. The neurotransmitter is glutamate, but there's other events and other chemicals that are involved. What I'd like to draw your attention to is that this long-term potentiation happens in terms of, well, here's the original connection where there's a nerve cell that's being stimulated, right? And it causes the release of glutamate. And here's some receptors for glutamate here. This red colored object represents a receptor. But notice that after long-term potentiation, other receptors have developed. So you get this, so you get a red receptor, you get the it's called upregulation, an increase in the number of receptors, not only the red one, but also other types that are able to participate in, neurotransmission. Right. So notice that here in this case, this is like in the early stages of the formation of this memory, this connection between nerve cells. And notice that there's only there's a couple of different receptor types here, and that there's two green ones and one red one. Over here, there's three green ones and two red ones. The point is that the cell has become more sensitive to the activity of the neurotransmitter. Now, what does this have to do with our course in perception? Well, what happens is that color, if an object is colored, it may trigger more of that more of that activity, so that we're able to develop a memory, a stronger memory for color. Why that happens, I don't know. We just seem to think that it does happen, right? So we know that color, seems to have a strong impact on memory for whatever reasons there are. Now, we've mentioned a couple of important concepts. We've discussed some observations about how the visual cortex is organized. And we saw the ocular dominance bands, and we've seen the orientation columns for different stimuli. We've seen some ideas about how the visual cortex is organized. We've mentioned the famous work of Hubel and Wiesel in terms of their experiments with kittens to show how the area on the cortex is expanded in an eye that is normally sensitive versus an eye that's sutured shut during the development of the kitten. We said a few words about the role of color in memory. And I'd like to mention a few concepts about the what are called the Gestalt laws or rules. In other words, rules of basic. So the Gestalt laws the Gestalt principles of organization. Here's a listing of the major Gestalt laws, at least some of them. The law of simplicity. In other words, every stimulus pattern is seen in such a way that the resulting structure is as simple as possible. So if you see something that looks like a cube, well, we see it as a cube. And then the law of similarity, similar objects to be grouped together, appear to be grouped together. The law of good continuation. In other words, we tend to see things in a reasonable flow pattern. Here's some examples, nice examples of some of these Gestalt principles. Law of similarity here. You may see two groups of dots. You see a red group and a black dot, or a green group, right? And like the jerseys that players on a football team wear. Well, you get one of the teams is wearing brown and the other is wearing blue. It's easy to determine which is which. Law of proximity here. Right. In other words, things that are grouped together tend to be seen as belonging together. So you have two groups of, columns of dots here. Right? We don't say, you might say, well, you see two groups of dots instead of just 6 columns, because these are close to each other and these are close to each other. See, law of pragnance, the law of good figure. Well, what do we see? Well, we probably see 5 circles. Kind of maybe the Olympics logo or something like that. The point is we're kind of taking some relatively complex shapes here. They don't have to be circles. They could be kind of oddly shaped figures, right? But it's pretty simple to see them as circles. Law of continuity, right? In other words, we see this as kind of a single line moving in a certain direction, or maybe no movement at all. Maybe just the idea that we're seeing it as a single object, The law of closure. The law of closure says that you may see a diamond here. There is no diamond, but we close it. We close this, the gaps, the spaces between the adjacent areas. The law of common region. You may say, well, you may perceive 2 dots belonging together here and 2 dots belonging together there because they belong to the same area in the picture. This could be pragnans down here, the law of good figure. In other words, it's reasonable for you to see something, maybe a cube here. In this case, you see another cube, right? But they're very similar. In fact, there is no cube here. There's a bunch of, kind of irregular patterns, but we kind of put them together in a meaningful configuration in our perceptual system. Over here, you may see, this is an important figure called figure and ground. It's a principle, I should say, called figure and ground. What 2 things might you see here? Well, you might see 2 people, two silhouettes of people, or you might see a music stand or a lectern. In other words, a podium or something. So that's so when you look at the podium or the music stand or whatever you perceive it as, well, notice that this is the figure ground principle. The figure kind of stands out against the background. So the figure is closer to you and the background is further away. Now, if you see the 2 faces facing each other, well, then the faces are closer to you. We tend to, as opposed to the background. So the background appears farther away. So, so we tend to see things in terms of, figure and ground. So those are several of these ideas that we have that kind of add up together into the the principles or the laws of Gestalt psychology. Gestalt is not a person. It's a set of principles. It's a school of psychology that says it's a good way to organize our perceptual experiences. And I'd like to say a few words about depth perception. Depth perception, the ability to perceive distance. Now, some of them are able to be carried out using pictures. Those are called the pictorial cues. And those are, those would include the monocular cues, the ones that work with just one eye, all right? So in other words, this is the topic of depth perception. So the pictorial cues, I should say, are the ones that, you can use one eye. In other words, you can look at a picture and you can see, you can perceive depth because of the way the artist has drawn, has produced the items in the picture. And you only need one eye for that. And they include occlusion, right? In other words, if an object covers up part of another object, it's probably closer to you. Relative height. Now, what does that mean? So it's like, well, the farther away an automobile gets, the smaller it looks on your retina, right? So in other words, if you look at two objects and one of them is farther away, actually that'd be like familiar size, right? So in other words, you don't think the car has actually gotten smaller. You have a knowledge that it's a distance away from you because of its size on our retina. We talk about relative height. We can say, well, gee, if there's a car over, a long distance away, and then there's a person next to the car, well, we can kind of judge, we can see those things and we can say, well, we know how big they're supposed to be. So we can judge them from, from our experience, basically. Some of these are kind of mixed together, like familiar size. So in other words, if you know how large a car is supposed to be, you can kind of estimate how far away it is. Atmospheric perspective is another pictorial cue or monocular cue. Objects that are less sharp appear to be at a greater distance from us. In other words, if you look at into the distance, you may see that things kind of tend to fade away. In other words, they tend to lose their sharpness. They kind of get a little bit dusky, in the distance. And of course, linear perspective, parallel lines converge, in the distance. In fact, let's take a look at some of these. These are, this might be what? These would be monocular depth cues using one eye. And notice that you might say, well, this may be the atmospheric, perspective, right? In other words, things get a little bit less clear in the distance. And of course, this is the familiar object thing. In other words, you have several cues going on here. You have, the familiar size. So this car back here looks, quite a distance away compared to the car in front. And of course, this one hides part of the one behind it, right? So if this car hides part of the one behind it, well, you can tell this one is a shorter distance away. Texture gradient. In other words, this is looks like a sun bleached arid landscape, right? So notice that it forms a texture gradient. So the gradient, the little pieces that form the gradient, the little pieces of the composition, they get smaller. So the ones that are close to you appear, they're quite a bit larger on your eye. But as you fade into the distance, they get small, like bricks on a brick road. Over here, you got the texture gradient effect. Right? So you got the bricks in the ground area here, and then they get smaller as they recede into the distance. And that the pool balls are all the same size. So as they cover up less of our retinal area in the distance. And then we have, of course, we have movement cues, such as motion parallax, right? So in other words, nearby objects seem to move past us rapidly. Reading from, distant objects appear to move more slowly. So if you're looking at two vehicles and they're both moving at 60 kilometers an hour, the one that's right next to you whizzes past really quickly. But the one that's, the one that's actually 300 meters away or 600, maybe a kilometer away, it tends to move. It looks like it's moving relatively slowly. That's called motion parallax, which we've discussed that earlier in our class. Those are the monocular depth cues. And in the binocular depth cues? Well, we have, we have what's called stereopsis, which is the use of two eyes to perceive, distance and depth. An important factor here is called retinal disparity, which means that, when you look at when you're using two eyes to see, in other words, you're not using just one eye, you're using two eyes. And two eyes each seeing a separate picture of the world. That's called retinal disparity, right? In other words, the idea being that when you look at something, you're actually getting kind of a different view of the same object, from the different eyes. So here, for example, here you're looking at something here, and notice that in the left eye sees a slightly different view of the same thing than the right eye does. You can do that by closing one eye after the other. In other words, look at the wall near you and close your right eye, and then close your left eye. And you'll notice that the objects you're looking at may appear to, they kind of look like they're moving, but the point is that you're getting a different view in each of the two eyes. That of course is how things like three-dimensional videos and comic books. You can look at something, you get a three-dimensional experience, even if you look, even if you're looking close up, because you can have a separate image projected to each eye, and you can combine them. Your mind kind of combines them. There's an important idea in this area called the horopter. The horopter is it's like a solid line that goes through your fixation point, right? So this person is fixated on this thing. In other words, the fixation point is just what you're looking at. That's all the fixation point is. So that the stimulation is coming into both eyes, on your two retinas. And if you draw an imaginary line through what you're looking at, that's the fixation point. And in fact, you see things in front of the fixation point in a phenomenon called crossed diplopia. Diplopia means double vision. And things that are beyond the horopter, you'd see in what's called uncrossed diplopia. Here's an interesting little experiment you can do. Take your forefinger or a couple of pencils or anything. Take your two fingers and hold them. Make one farther away. Make one maybe 20 centimeters farther away than the near finger. And what you can do is, if you focus on the far finger, you're looking at the far finger away. You see kind of a double image of your finger that's right in front of that's closer to you. Now, if you keep looking at the far finger or the far pencil, whatever it is you're holding up, right? And keep looking at that far finger. And if you close your eye, close your left eye, you'll find that the right ghost image of the near finger disappears. And similarly, if you close your right eye, then the left image of the near finger disappears. On the other hand, if you focus on the near finger, the near object, right? There's a horopter that goes, your horopter is moved to the object that you're looking at. If you close your left eye, then you'll find that the left ghost image beyond your fixation point disappears, right? So that's the idea. You can have, the idea is, and that's due to the different images, two different images falling on the different eyes when we look at the same object, with two eyes. There's also this concept of, the oculomotor cues that we have, right? In other words, when we have, things, parts of your visual system literally move. And notice, in other words, when you look at an object, when it's close to you, as you move your finger, put your finger at arm's length, and then if you move your finger toward your head, well, your eyes kind of converge. That's called, convergence, as an object is moved closer to you. As they move farther away, well, then your eyes, diverge, right? Like on the right side here. So to a distant object. And convergent for a normal eye, in other words, you don't have nearsightedness or farsightedness. This convergence begins to happen when an object is closer than 6 meters away. If you're nearsighted or farsighted, then the idea is your lens isn't focusing well, and it had happened at a different distance. But notice that one of the two oculomotor cues is called convergence of the eye, convergence, and the other is called accommodation. You may recall we've mentioned accommodation as the as the thickening of the lens. This is the lens over here, right behind the pupil of your eye, the opening in the front of your eye. And what happens is that lens gets thicker as an object is moved closer, because it has more focusing to do to put the image onto the back of the retina here. So those are called the oculomotor cues, right? Oculomotor means eye movement. Cues based on eye position and eye muscle tension. They include convergence. So we have what are called binocular cues to vision. And in other words, if you're undergoing convergence, then you know an object probably at an unconscious level. You can tell that they kind of add to your appreciation and your perception of depth. So we have, of course, that would only work, and in a close distance, less than 6 meters away. But certainly, if you're looking at an object farther away than 6 meters, you're looking at something 300 meters away. You're not converging at all. So your eyes are just totally not in a state of convergence. They're totally diverged. So those are a few words about, depth perception. We have different cues. And of course, we have, in the brain, recall that the brain, has the dorsal and ventral stream, right? So the dorsal stream handles vision movement cues, right? So movement and vision is kind of processed. It's one of those, we mentioned that we can remember the concept of feature detection, where we looked at the picture of the flower a little earlier, right? And we can say, well, gee, there are different features going on in the flower. And we mentioned what form and color and movement, right? Motion is one of those things. Motion, so in other words, distance is something that motion helps to show us, including motion parallax. So we'd say that movement is one of those features in the that we undergo, that we can appreciate in the processes of feature, the process of feature detection. Now, visual illusions, particularly some of the the size illusions are pretty interesting. We've mentioned other kinds of illusions like the Mach band effect. That's MACH. In other words, we that are due to that. We mentioned lateral inhibition responsible for that. But visual illusions are some of them are very common. The Müller-Lyer illusion is famous, right? In other words, your tendency, if it works for you, and this was discussed in one of the early videos that we had in our course, which is, if the illusion works for you, you'd tend to see the bottom line here. Take out the fins, and you'd see the line as longer than the one on the top, when in fact they're basically the same. Various explanations for that. One of them would be that the additional fins going outward would tend to give the kind of the overall impression of a larger size, a longer size. And then the other interpretation would be that we'd see it as kind of moving toward us. We'll get into the ideas that if an object appears farther away, this one appears farther away. If it's like in the corner of a room, if you can use that analogy in your thinking style, things that are farther away should get smaller, but in fact they don't. In some of these illusions, for example, the famous railroad track illusion, or the Ponzo illusion, as it's called. Notice that if the Ponzo illusion works for you, then you may see that the blue bar, the bar that's higher up in the picture appears larger if the Ponzo illusion works for you. Here's a few more examples of that. Due to the linear perspective idea. So it's usually a pretty strong illusion that seems to work pretty well for lots of people. There's a couple of reasons that the top line or bar should appear larger. One is that it covers more of the texture gradient. Right. In other words, the railroad tracks, the trestle, the railroad ties. There's more texture covered, and therefore it has the impression of being a larger object. The bar does, and plus it's farther away. If it's, in other words, it's and plus it's higher up in the field of view. We may have mentioned. I'm not sure we did. If you look at a picture, if something is higher in the picture, it looks farther away. So if this is a picture, well, we can say that I guess this is a train here. This train appears a lot farther away than the near section of the railroad here, because it's higher up, physically higher in the picture. And notice that this bar is physically higher up in the picture. That's another reason. Not only the texture gradient that it covers is more, but also it just looks farther away. And if it's farther away, see, it's all psychological. It's farther away. It should look smaller if it's the same size. See, that's the key. If it's the same size as the bar in front, it should look smaller if it's farther away. So there's distance cues, there's depth cues there. So we get the illusion that it must be bigger if it's farther away and it covers the same retinal area. The same thing with this, the famous moon illusion, right? The moon illusion that, maybe you've experienced it, but the moon looks a lot larger on the horizon than it does when it's up in the middle of the sky, right? And the reason for that, as far as we know, and people have done lots of studies of this moon illusion, that the concept is that when it's on the horizon, in other words, the moon is just coming up, as we say. And you can see it coming up, and it's lighted, strongly. Well, guess what? People think it looks farther away. Because why? Because we have depth cues, we have texture gradients. Look at the gradients in the rocks, the land here, and look at the distance cues provided by the converging lines of the railroad track. We have mountains. And when you look out in a normal, moon coming up, right? Well, you might have buildings and houses and, not just mountains, but all kinds of things in your environment to suggest, hey, that moon's pretty far away. But guess what? When people look at the what's called the arc of the heavens, when people look at the moon that's straight up at the sky, we tend to see it as not so far away. It's an odd thing. It's called the arc of the heavens phenomenon. So if you look at the moon, a lot of people would have the impression that the moon that's up in the sky, straight up, right? In other words, you're kind of looking up. You're not looking on the horizon. You're looking at the moon in the middle of the sky. Well, there really are no depth cues up there. There may be a couple of clouds, but beyond that, there aren't really depth cues. And people tend to have the impression the moon actually looks closer, believe it or not, if it's straight up in the sky, as opposed to on the horizon. See, when the moon is on the horizon, it tends to look farther away, at least for most people, as far as I know. And therefore, if it's farther away, but it covers the same retinal area as the moon in the sky, and therefore it must be larger. See, it's all psychological. Farther away, same retinal area. We say it's subtended, it subtends or takes up the same retinal area. It must be larger. So it's all psychological. The moon illusion is. So those are a couple of illusions, particularly illusions of depth and distance. So they're basically situations where size constancy breaks down. Maybe you've seen the famous Ames room. where people are able to, appear different size based on their distance. Oop, got to see that. Can't see that one right at the moment here. There's the Ames room, right? So you'll see that if the effect works for you, you might say, gee whiz, look at the size of this person here and this size of this person. Well, guess what? The Ames room is actually built in a special way. In other words, the floor isn't really rectangular. The person who's on the right side here is actually very, very much closer to us than the other person. And therefore, and what happened is that the people who built the room arranged the texture gradient on the floor, for example, to be to be able to provide the illusion that it is a regularly shaped room, when in fact the person on the left is quite a bit farther away than the person on the right. So that, in other words, we have familiar size at work here. So we have this kind of a situation. These depth Q illusions are situations where we're kind of tricked by the broad, the big context in which we see something. So these are illusions of, illusions of depth and distance. So those are some thoughts about how vision is decoded. We mentioned feature detection. We said a few words about, the kinds of neural events that are happening in the visual part of the brain when we perceive something. We looked at the linkage between color and memory, and we examined some of the famous Gestalt principles and remember the binocular and monocular cues for depth and distance. And we finally ended up examining some of the famous illusions of depth. Good, thank you.